goes behind or in front. When you swap the two eyes, yeah, and swap the two eyes, then the depths will change. And this is an example of the topic we are going to talk today. We're going to teach a new topic today, finally visual recognition. They say, you're studying vision, this vision. How come the whole time we never talk about recognition? Because there's so many hidden jobs to do before the recognition. The brain just goes crazy, yeah. So, too far or not? Is the effect okay still from that angle? Yes, good. Okay, so today we'll talk about recognition, yeah, with this a prelude. So today I'm better with my voice, so I'll try to have a mixture of talking and a little composition of videos. So today, after the selection, we are going to talk about statistical maximum likelihood decoding, which is called recognition. Sometimes people call it Bayesian. Tomorrow we have a more modern topic. Okay, so.
Visual decoding is often called visual recognition or visual inference. Let's start with some introduction and definitions. Visual decoding is the third of the three stages of vision: encoding, selection, and decoding. So it's after visual inputs are sampled, represented in neural responses, and after a fraction of these inputs is attentionally selected for further processing. It infers the properties S of a visual scene from neural responses R. The inferred properties of the visual inputs are therefore perceived. Among the three stages of vision in the brain, decoding is the least known and least understood, particularly in terms of linking neural responses to visual behavior. The properties S may be a face identity, the color of a surface, the orientation or the motion direction of an object, etc. Due to attentional selection, not every property of the visual scene is decoded. For example, you may recognize the foreground object, but not the background objects. You may recognize a face identity, but not the facial emotion. Or you may recognize the motion of a tiger, but not which specific tiger it is. Let's see a demonstration that we do not decode much of visual inputs due to our attentional selection. In attentional blindness, can you tell the difference between these two images? We have the impression that we see all the big and small details in these images clearly. If so, you should be able to tell the difference between these two images right away. But many people cannot spot a very big difference within tens of seconds. But once I point this out to you, it's obvious. So this demonstrates that we are blind to most things in front of us. Our vision is constrained by an attentional or information processing bottleneck, such that we do not see most of visual inputs in front of us unless it's selected into our attentional bottleneck. In this framework of vision as three stages, the neural activities R used for decoding should be those representing visual inputs that are already selected by attention. Exactly what R is, we know very little. except to assume that they are in the neural activities in animals' brain, such as in monkeys' visual cortices. In practice, researchers often try to decode from their own choices of representation R. For example, R can be retinal or V1 responses to visual inputs, often because they are better known than other brain responses. Or R can be extra striate responses to visual inputs, or can be pixel values in visual images in computer vision practices, for example. And R can also be functional imaging and EEG measurements, sometimes used for brain-computer interface. These decoding practices at least serve the purpose of seeing what information is available in the representations R. Let's look at some remarkable observations about visual recognition in humans. Let's see a random dot stereogram. Here is a square array of random black and white dots. We add a smaller square array of random dots on top of it. Now you cannot see where the smaller square is because the two squares have the same randomness for their dots. We use this image as the visual input to the left eye and make a copy of it to the right eye. These are the borders of the smaller square, but without the highlight you cannot see these squares. Now if I move the small square in the left eye image a little to the right, once you fuse the two molecular images in your brain, the small square should appear in front of the big square. The brain can work out the correspondence between the two molecular images to identify this foreground square separately from the background square, even though each molecular image gives no clue of the small square.
Try, yeah? You have already tried, for those of you who arrived earlier, because we just put one image to the left eye, one image to the right eye. And if you just close one eye, you could not tell there is a disc. But if you open both eyes, you see there's a disc in front or behind, depending on which eye sees the blue. Okay, that's just an example. So this is an example where your brain decode for the depths and decode for the presence of the disk using just two random dot images. Yeah, you can say you can use these two random dot image and do the decoding. That's an example. Okay, so let's see the next example. Oh, sorry, what happened?
If you fixate your gaze here, you can still see the letter T to the left and to the right without shifting your gaze to them. This means that although your peripheral vision away from your gaze at this distance has a lower visual acuity, this acuity is still good enough for you to recognize the letter. However, if you add 4 surrounding letters as neighbors to this letter T to the left, keep your gaze still on the central cross. Somehow you cannot see this crowded T now, even though each neighboring letter is not overlapping with the letter T in the center of the crowd. This is visual crowding caused by something in the brain, not in the eye. If you move your gaze to it, now you can see the letter T clearly. So crowding is present in peripheral vision, but not in central vision. In this image, A is a dark square, B is a lighter square, but in fact they have the same shade for their image pixels, perhaps more convincing to verify this way. Square B is in a shadow. The brain infers that it is lighter. In this image, you should be able to see a vivid white triangle here. But in fact, the only hints to this triangle come from these contour segments and line endings associated with the 3 black discs and this second triangle. The brain infers this white triangle so vividly that its white surface appears almost whiter than the white background. The brain also infers the 3 black discs and the second triangle from the occlusion by the inferred white triangle in front of them. Here you see fragments, and it's unclear what they represent. Here is another version with the same fragments, but behind some occluders. We immediately recognize that behind the occluders are 5 letters B. What is in this image? It is perhaps difficult to recognize. However, after you are given a hint, now your brain can see the duck clearly without the hint. Let's have a look at the object invariance in visual object recognition. We can recognize an object to be the same object when the image of this object changes. These changes are caused by changing the direction to look at the object, changes in object location, in lighting, or in distance between the viewer and the object. Note that when an object changes its location, one can always move our eyes to place the object in our central visual field. Let's demonstrate that object invariance occurs after visual selection. For example, in this image, we have a unique right-tilted bar among left-tilted bars. If we superpose a vertical or a horizontal bar to each of these bars, we get this image. Now it's harder to find that unique oblique bar. It's still at the same location, but somehow much harder to find now. Why? We notice that in various visual cortical areas, V1 has neurons tuned to various orientations of these bars, and it guides attention to the uniquely tilted bar because this bar is salient by its unique orientation. This stage is visual selection by V1. However, higher cortical areas, including IT, are involved in object recognition. They recognize this X-shaped object made of 2 intersecting bars, and also these neighboring X-shaped objects. Due to object invariance, even though these different X-shaped objects have different orientations, they are seen as having the same shape. So while this bar is uniquely tilted, this X-shaped object is no longer unique. This confuses us. Next, we show you that this confusion, due to object invariance, occurs after our gaze shifts to this object. I'll show you a larger version of the previous image. Let's find a uniquely right-tilted bar in it. Ready? Go! Most people find it hard to find it in their first try. Have you found it after a few seconds? Here it is. The blue trace is the gaze trajectory of one observer in visual search for this target. The gaze starts from the central point before this image is shown, and then after this image appears, the gaze arrives at the target very quickly. For most people, their gaze arrives at the target within 2 to 3 seconds.
So for most of you, you already move your eyes to it. But how come none of you see it? No? Anybody saw it before I showed? Raise your hand. Only one person. Okay? So that means this is a kind of involuntary, you did not know. V1 did it for you. Okay? In fact, statistically, 50% of the trials for most people, that means they never train, they never try this, but first time. 50% of trials, their eye go to it in 1 second. by 3 seconds, most likely you're already there. Okay, so therefore, it's amazing our brain can do a lot of this fast stuff that you did not know.
The fact that you tell what the task is makes search more complicated. Say, if you don't say what the task is and just present this, maybe the gaze would go there quicker.
Also, yes. That I haven't tried, but the gaze goes there already quickly. Yeah, as I say, within 1 second, 50% of child, they already went there. Within 2 seconds, 70% child, their gaze already went there. Within 3 seconds, 90% of child. So therefore, but 3 seconds, only one person, yes, 4 seconds. So the idea is, you can see afterwards, yeah.
This is visual selection of this target location, mainly guided by the saliency of the uniquely oriented bar. Then, after the gaze arrives, the gaze trajectory continues following this red-colored trace. The gaze hesitates around the target, then abandons the target to search elsewhere before eventually returning to the target.
This happened to almost
This is
all
likely why most of us did not realize that our gaze had visited the target quite a while back before we finally identified the target. This demonstrates that object recognition and object invariance occur after visual selection of the object location. If we fixate our gaze here at the center, it's very difficult to recognize this target by our peripheral vision. Our gaze went towards it, not because this object was recognized, but because this location is salient to attract our attentional selection. Now let's look at the neural basis for visual decoding or recognition. As mentioned, among encoding, selection, and decoding, we know the least about decoding in terms of its neural basis. Along the visual pathway, since much evidence suggests that selection starts in V1, decoding likely involves cooperation between V1 and extrastriate cortical areas. From the lesion effects we have already talked about, it looks like V2 and V4 are involved in decoding. Here are some example observations that V2 neural responses are associated with our perception or outcomes from visual decoding. Here, A, B, C, D, E, F, G are 7 patterns of visual inputs, and this red oval in each pattern is not part of these inputs, but marks the location of the residual field of a V2 neuron. This neuron responds to a vertical luminance edge or bar in input patterns A and D. In the other 5 patterns, B, C, E, F, G, the input pattern contains no vertical bar or edge inside the residual field. However, the neuron still responds in B, E, and F, but not in C or in G. This is because visual decoding lets us see a rectangle here in B. Its right vertical edge passes through the residual field, and a vertical texture border here in E, and a white bar extending here in F, but no border or edge is perceived inside the residual field in C or in G. So therefore, this V2 neuron's response corresponds to our perception, which is the outcome of visual decoding. And this is regardless of whether our perception arises from an explicit vertical edge or bar, or just implicit or inferred ones. A V2 neuron's response can also depend on whether a border belongs to one or another perceived surface. For example, in H and in J, the same image pixels are within the residual field in these 2 cases, and these pixels show a border between a white region and a dark region. The border in H belongs to a perceived bright square to its left, but the border in J belongs to a perceived dark square to its right. And this V2 neuron may respond more in H than in J. Such a neuron signals border ownership regarding which surface owns a border. When this neuron is exposed to visual inputs in I and K, it will respond more in I than in K, because in I, this border belongs to a surface to its left, but in K, it belongs to a surface to its right. Many V4 neurons are also tuned to border ownership. Compared to neurons in V1 and V2, V4 neural responses are more correlated with visual perception rather than retinal inputs. For example, during binocular rivalry, when one image is shown to the left eye and another image is shown to the right eye, we sometimes see only the image in the left eye and sometimes only see the image in the right eye. This perceptual switch is correlated with switches in V4 neural responses, while the retinal inputs stay unchanged. Beyond V1, in V2, V4, and IT, many neurons are tuned to image shapes resembling object components. Some cortical patches in temporal cortex consist almost entirely of neurons selected for face-like inputs. Here is how an example neuron's responses change with changes in facial features, such as facial aspect ratio or face direction, etc. Here it increases its response when the size of the iris increases or when the height of facial feature assembly decreases. After the onset of a visual input, V1 neurons start to respond about 40 milliseconds later. For V4, it takes about 70 milliseconds before they start to respond. And this is 100 milliseconds for IT neurons. By 125 milliseconds, responses from 300 IT neurons can be used as a population to classify visual input objects into categories by a linear classifier. However, when object discrimination is harder because, say, some parts of the objects are occluded in the visual inputs or are deformed, then we need to wait for later parts of IT neural responses to use them in a linear classifier to classify objects sufficiently well. And this is both in monkeys and humans. And this temporal delay suggests the involvement of both feed-forward and feedback processes in the brain's neural circuit to achieve more informative neural responses. Let's introduce maximum likelihood decoding from responses of a single neuron. Here is an example. Let's say a visual input contains many moving dots moving in different directions. In this figure, we visualize the motion direction of each dot by an arrow. And a monkey looking at this input scene needs to report whether the overall or average motion direction is upward or downward. In this example, the correct answer should be upward motion. So let's describe this visual input scene property, the motion direction, by S. S has two possible values, S equal to up or down. The monkey has to rely on the responses R of its neurons to do the task. Let's focus on only one neuron. Its response property can be described by how likely each response R is in different input conditions. When the overall motion is in the preferred direction of this neuron, let's say this is the probability distribution of possible responses R. Its average response R is roughly here. When the overall motion is in the opposite direction, then its responses are on average weaker. So this motion direction is this neuron's non-preferred motion direction. Decoding is to use the neural response value R to decide on what S is. Let's denote this decided value S as S hat. S hat may be different from the original S. When the neural response R is very high, then this strong response is quite certainly suggesting that the motion direction in this input is the preferred direction of this neuron, for example, the upward motion. And this should be the value of S hat, very likely a correct answer. Similarly, a very low response is quite certainly suggesting the opposite motion direction. However, when the response is not high enough in this example, then it's uncertain. From this neuron's response property, the preferred motion direction, upward in our example, is more likely the correct answer, if the original S is equally likely to be upward or downward a priori. Similarly, this lower response should make a better choice of S hat being the downward direction. The maximum likelihood decoding is to choose the S hat value to maximize the likelihood that a response value R is observed. With this R as an example, this is the likelihood that this response R occurs if the visual motion is in the preferred motion direction. And this is the likelihood that this response R occurs if the motion is in the other direction. So the likelihood is higher for the preferred motion direction to give this decision. Therefore, we can find a decision boundary as the threshold value of the neural response R. When the response is larger than this threshold, the best decision is the preferred motion direction. Otherwise, the best decision is the non-preferred direction. The size of this gray overlapping area between the two curves scales with the probability of error that this optimal choice S hat is incorrect. Pulling the two curves apart reduces this overlap, or the error rate. When these two curves are Gaussian, the distance d between the two peaks divided by the standard deviation of the Gaussians defines a measure d prime, a discrimination index, for the sensitivity to discriminate between the two visual motion directions by this neuron's response R. This makes sense, since when the two curves are more apart from each other, the neural response values for the two motion directions are on average more different from each other. In such a case, in the range of all possible neural responses R, when this response R is within these two large subranges, the maximum likelihood decision for the motion direction is almost certainly correct. When these two curves are further apart from each other, the accuracy of the decoding performance is higher. This occurs when the dots are moving more coherently, so that more percentage of dots are moving along the overall motion direction. This is the decoding accuracy versus the motion coherence by decoding from a neuron's response in monkey's visual cortical area MT. The accuracy increases with the coherence of motion. Surprisingly, this decoding accuracy from the response of one NT neuron is comparable to the performance of the monkey doing this task of judging the overall motion coherence. And this monkey's MT should have millions of neurons. From the likelihood function, the conditional probability of seeing feature value S given the neural response R is given by the Bayesian formula as this. Here PS is the prior probability of S, and this is the marginal or overall probability of response R. Then a related decoding scheme is to choose S hat so as to maximize this conditional probability of S given R. This decoding scheme is often referred to as Bayesian decoding. Therefore, maximum likelihood decoding is a special case of this scheme when there is no prior bias for any S. And this is the case in our example when the upward motion and downward motion are a priori equally likely to occur in the visual scene. Let's look at the maximum likelihood decoding from responses from a population of neurons. Let's recall that when a visual input S is encoded by one neuron's response R, the probability distributions of responses R are like this for input A versus B. For a given response R in any trial, this is the likelihood of this response if the input is from A, and this is if the input is B. The maximum likelihood decoding decides that the input is A or B depending on which choice has the higher likelihood. In this example, the likelihood from A is higher than that from B, or equivalently, the ratio between these two likelihoods is larger than 1, or the log of this ratio is larger than 0. So therefore, the decoded visual input is A. If we have a population of neurons, the response R is now a vector with components R1, R2, R3, etc. from neurons 1, 2, 3, etc. And then to decode whether the input is A or B, we look at this likelihood ratio, where the response R is this vector. And we see whether this ratio is larger or smaller than 1, or whether this logarithm of the ratio is larger or smaller than 0. When the responses from different neurons are conditionally independent of each other, once the input S is given, whether this input is A or B, then the probability factorizes, meaning that the probability of the whole vector R is equal to the multiplication of the probabilities for each response components R1, R2, R3, etc. Then this log ratio becomes the summation of log ratios for each neuron, 1, 2, 3, etc. And decoding will be based on this value. In the brain, a neuron's response is often approximately Poisson, with its distribution given by this formula. For example, if the input is A, this will be the distribution of response Ri from neuron I. And this expression R bar is the average of the response Ri in this distribution. And this average also appears here in this mathematical formula. And analogously, when the input is B, then you can work out the math to see that this log of the likelihood ratio follows this expression for response Ri. Consequently, the decoding decision should be based on this inequality. Notice that this expression depends only here on the stochastic response Ri of various neurons. And these other quantities are just parameters fixed from the neural properties, namely the average neural responses to the two visual inputs, A and B. Then this is a linear weighted sum of responses, and this is a decision variable, while this is the decision boundary for the decoding to decide from this population neural responses whether the visual input is A or B. Let's use an example to decode whether the retinal input is A or B based on photoreceptor responses. The population response factor to input A is this one on average, and is this one when the input is B. Then, given a population response in the photoreceptors, the decision variable can be constructed by such a linear network whose output, capital R, is the linear weighted sum of photoreceptor responses R1, R2, etc. The weights in this network are the weights in our decision variable. The probability distribution of this decision variable R is like this when the input is A and like this when the input is B. Its mean value is here when the input is A, and the mean value is here when the input is B. And the decision boundary is here, so that when we have a photoreceptor population response pattern, when this decision variable, capital R, is here to the right or here to the left of this decision boundary, the best guess for the retinal input is A or B respectively, according to the maximum likelihood decoding method. Let's look at this distance d, and then the discrimination index d prime. D follows this.
The d is distance between these two, and somehow it disappeared.
A or B based on photoreceptor responses. The population response factor to input A is this one on average, and is this one when the input is B. Then the probability distribution of this decision variable R is like this when the input is A, and like this when the input is B. Its mean value is here when the input is. Let's look at this distance D, and then the discrimination index D prime. D follows this expression as the difference between these two mean values. So it grows roughly linearly with the number of neurons in the population. The variance of decision variable R is the summation of the variances from the individual responses, of course weighted appropriately. This is because the neural response noises are independent of each other. This is the assumption, yeah. And this means the standard deviation of this capital R grows only sublinearly, roughly with the square root of the number of neurons. And plugging these in here, this means that d prime of the whole population increases with the neural population size, growing roughly sublinearly with the number of neurons. Approximately, this is how the d prime of the whole population relates to the d prime from the individual neurons, I. This means that the maximum likelihood decoding accuracy should increase with the size of the neural population. It is accordingly surprising that the decoding accuracy from the responses of a single empty neuron is roughly the same as that by the whole monkey with millions of empty neurons. This suggests a processing bottleneck in the brain. Let's look at two example applications of maximum likelihood decoding to understand visual contrast detection and discrimination. Such a method has been applied to a contrast detection task. Human observers see a display that is either a blank or has a grating. Each image is shown for only 100 milliseconds, and these two images have the same average luminance. The human observer has a task to report whether the image
So maybe I'll just stop here to ask for questions for a while, yeah, before we continue. Questions? Yeah, okay. You mean the monkeys? Monkeys running? Okay, yeah, yeah. Oh, I see. It's no. Good question. Okay. Most people don't do that. Okay. Okay. Because a neuron prefers orientation. Neuron also prefers motion direction. That's talked about in efficient coding. Remember, we have neurons prefer this color, that orientation, and motion. And so in MT, you also have neuron prefer this direction or that direction because they're tuned to direction, motion direction. Okay. Yeah. That's right. It's as good as the whole monkey. Yes, because you just wonder, how does this monkey do the task just relying on one neuron? That doesn't make sense, yeah? And so, so that's why you think that something is wrong. And this something is the bottleneck. The brain just does not use all the neurons. So for example, here, motion strength. Okay, motion strength means how much is signal, how much is noise. So this has mostly a lot of noise. So 100% of dots moving in this way, it means very strong motion, clear signal, yeah? Okay, and this neuron, monkey can do roughly about, by the time it's 10% of dots moving in the right direction, the other 90% all noise. The monkey is already doing 75% correct. Because each time monkey has a motion up and motion down. If the monkey completely guessing, only 50% chance correct. Okay, so there's 50% chance here when there's very weak motion. But by the time you have 10% of dots moving, it's already 75% correct. That's how the monkey does. However, when you do the experiment, this experimenter is doing the experiment. He's going to use the response of this neuron and see whether it's larger or smaller than this threshold, and also make a decision. And then also can calculate based on that decision how often it's correct. And you find it's also 75% correct. So the experimenter just use one neuron from this monkey is enough to make the same percentage of correct guess as the monkey. That's what I mean. Okay. And then this is related to here. The more neurons you have, the better guess you should have. So why is it the case that the monkey is not using? Monkey is not doing it. Okay, that's the question. So the brain is not able to do it by some processing bottleneck. That's the idea. Okay. Any more questions? Okay. Associated with maximum probability of. Okay. So it's here. So imagine you have a response here. Okay, let's say this response is 70 spikes per second, let's see. Then say response here, if the input is A, then this is the probability, this should be the response here. Probability, let's say, is 10%. I don't know. But if input is B, that's only probability 5%, it's going to be here. So the probability for A is larger than probability for B. That's what it is. And this probability is called likelihood. See how likely you'll get this response. That's right. So this is maximum likelihood decoding. But this time you say it's A rather than B, assuming A and B are equally likely, and let's say each time it could be E, A, or B equal. But what if in an experiment, it's 90% time it's moving upwards, 10% moving downwards. So if you're unsure, you're going to go upwards, right? So you will also have a prior, and that's a Bayesian decoding. So the idea is, if you, where's the Bayesian decoding? Did I just lose it? Have you gone through? Yeah, Bayesian decoding is probability. What the S is given R is the probability. What R is given S. The prior probability. This is the prior. You should decode according to this. This is, of course, divided by PR. But given R, this is fixed. Given R, this is already fixed. So therefore, if this you don't care, then you decode according to this is equivalent to decode according to this, whatever is bigger. So you want, yeah? So this is called likelihood. And this is, if you decode by this, you say, if S equal to A bigger or S equal to B bigger, according to this, it's called maximum likelihood decoding. But if you decode according to this, whether S is equal to A or B, according to this, which one is bigger, then you are decoding by Bayesian decoding because you are also counting that. Okay, yeah. This is just mathematics. There is applying mathematics to data, yeah. Whenever you have something, you say, what is the best, most likely? You always decode by this. It's just in a special case, okay? You say, given R, what is S? What is that question? Whenever you have that question, you say, what is the probability what S is given R? You always decode based on this because that's the mathematical expression of this question. So it's not an assumption. It's just if you don't use this, it's always less optimal because that's always the best. In this special case, decode like this is equivalent to decode like that. That's what. And then we apply to neuroscience. It doesn't have to be neuroscience. It can be engineering. It can be brain-computer interface, anything you want. Yeah, it's always like this. Okay, good. So you have a space of X, okay? You, in the space of X, find the maximum. But if the space of X has only two S values, you just choose the bigger one. But if the space of X here, the S only take two values, A or B. But later, S could be 100 values, could be multidimensional. Yeah. Here we are just having only two values for teaching. Okay. So imagine you say, I want to decode how tall I am. And tall can be, then it's many. Okay, so imagine you have a space of X. And you already given R is the value you have. Then you have S given R. And in this, you just choose whatever is the maximum. Okay, then this S should be S hat. This, in the space of X, choose the maximum. Okay? But if the space X has only two values, then you just choose the higher one. Yeah? Okay, yeah, good. So let's apply it to a real case.
To a contrast detection task. Human observers see a display that is either a blank or has a grating. Each image is shown for only 100 milliseconds, and these two images have the same average luminance. The human observer has a task to report whether the image shown is A or B. The image contrast for the grating is weak, so the task is really to detect this contrast, whether it's present or not. Many experiments are done in a psychophysical paradigm called a two-interval forced choice. Visual inputs A and B are shown in two separate time intervals, interval 1 and interval 2. And the observer, after seeing the two images, reports whether input A was in interval 1 or 2. So with the stochastic decision variable R, let's say that its value is R1 for the first interval and R2 for the second interval, then the maximum likelihood decision should be, for example, to report pattern A in interval 1 or 2, based on whether R1 minus R2 is positive or not. From what is known about photoreceptors, one can calculate the average responses of each photoreceptor to these two images to give the average distance between R1 and R2 and the variance of this distance. Therefore, we have this d prime for the decoding, and this in turn gives the decoding accuracy, which is the probability that the maximum likelihood decision A or B in the first interval is correct. Since this d prime value depends on the grating's contrast C, one can calculate the threshold contrast value delta C that is needed to get this accuracy to a threshold value, say 70%, and compare this threshold value obtained from this maximum likelihood decoding method with that by the human observers, that is the actual contrast threshold value needed by the human observers to do their task with this accuracy. This is the result. The vertical axis is the contrast sensitivity as the inverse of the threshold contrast value, delta C. The horizontal axis marks the spatial frequency of the grating in this image. The curves are predicted from the maximum likelihood decoding. The data points are from the human observer's behavior. And the good news is that data matches the prediction well, particularly in how sensitivities decrease with increasing spatial frequencies of the gratings and increase with the background light levels, indicated here for 3 light levels. The bad news is that, in fact, this match between data and prediction is only achieved by scaling down the predicted values by a large factor. This means that the human observer performance is much worse than what is theoretically possible, even though house sensitivity should decrease with the spatial frequency or increase with the light level are just as predicted. This again implies that there is a bottleneck in brain's processing power. In the literature, what's theoretically possible is called the behavior of an ideal observer, and the analysis to get this theoretical possibility is called the ideal observer analysis. In another example, we can find the minimum contrast difference needed to identify which of the two images has a stronger contrast. Let's compare the prediction from the ideal observer analysis with data obtained from human and monkey observers. This is the contrast discrimination threshold, delta C, versus the baseline contrast value, C. The data points are as from these human or monkey observers, and the curve shows the ideal observer's performance using responses of only 71 V1 neurons. And since V1 has millions of neurons, this result shows again that the real observers are far worse than the ideal observer if responses from all V1 neurons are used for this maximum likelihood decoding. Let's apply maximum likelihood decoding to understand color discrimination from cone responses. We have shown that photoreceptor responses can be used to decide whether the retinal input is A or B of two given possibilities by a decision based on whether A or B gives a higher likelihood for the observed population responses in the photoreceptors. Now let's show how to distinguish a color input from similar inputs in a two-dimensional input feature space using maximum likelihood decoding. Visual inputs are restricted here to be monochromatic lights described by two feature values, wavelength lambda and light intensity I. Here are two color patches that appear different from each other. What is the minimum or threshold difference, delta lambda, needed to distinguish two color patches? However, in this case, do these two patches differ in wavelength lambda or light intensity I? For example, is the right patch simply having a weaker intensity? Or perhaps they differ in both lambda and I. In the face of such a confound between lambda and I, one experiment gave wavelength lambda to one color patch, the reference field, and lambda plus d lambda to the other color patch, the test field. When d lambda is sufficiently small, human observers can adjust the intensity of the test field to make the two patches appear identical to each other. However, when d lambda is too large, this is impossible. No matter how one adjusts the intensity, the two patches appear different from each other. Threshold delta lambda is the smallest d lambda when this impossibility occurs. This is how this threshold depends on the wavelength lambda. Let's understand this non-trivial data by maximum likelihood decoding. These are the spectral sensitivities of the red, green, and blue cones as functions of lambda, and these cones are also called long, median, and short wavelength cones respectively, and this is their responses to monochromatic lights. In the three-dimensional space of responses from the three cone types, this is, for example, a distribution of possible responses to wavelength lambda at a particular light intensity, and this is the distribution for a similar wavelength. When these two distributions overlap more, then it is more difficult to perceptually tell any difference between these two inputs. This is the mean response to a given input for each cone type, long, median, or short. And here this function is the effective spectral sensitivity curve made from the cone density NA, so we are pulling the local cones of the same cone type together to form an effective giant single cone. And this is the pre-receptor absorption factor, OA, to account for the light absorption in the eyeball before the light reaches the cones. And this is the cone fundamental, which is the spectral sensitivity function of a single cone before pulling the local cones of the same cone type together. All these three factors are experimentally known quantities, and of course, the mean response scales with light intensity I and the effective sensitivity. Each cone's response or absorption RA follows a Poisson distribution like this, and these are the mean absorptions. And since the three cones are assumed to respond independently, their joint probability is the multiplication of the individual probabilities and to give this result. And this joint probability is the likelihood of the responses. And therefore, this is the logarithm of the likelihood, and it's a summation over the log likelihood from individual cone types. Given this log likelihood, the maximum likelihood solution for input features lambda and I is the solution to.
So here's the example, yeah? So now you need to decode for this S. S is now a two-dimensional continuous variable. You're still trying to find the largest S, okay? You say, what is the S? What is the S value, not largest? The S value that make this largest, okay? And that's why you do a derivative, trying to find where the largest is. So what is it going on? Okay, sorry.
likelihood solution for input features lambda and I is the solution to these two equations. This equation makes the likelihood peak in the feature dimension of light intensity I, and this makes it peak in the dimension for wavelength lambda. One can make a contour plot of the likelihood near and around the peak in the two-dimensional input feature space along wavelength lambda and light intensity I, centered at the peak likelihood. For example, along this contour is a constant likelihood, which is near the peak likelihood value, but slightly smaller than the peak likelihood. And along this contour is a next slightly smaller, lower likelihood, and so on. And we define this contour as where the likelihood drops to a threshold value, say 50% of the peak likelihood, so that all these feature values within this closed region are almost equally likely, so that they are perceptually indistinguishable from each other. This also means that we can approximate the distribution of the most likely feature as a Gaussian distribution centered on the true actual input feature. And the shape of this distribution comes from the correlation structure defined here as the Fisher information in the two-dimensional space. Its values are characterized by the second derivatives of the log likelihood function from above. When the lambda value is not far from the center, then observers can find this range of input intensity to make the color patch indistinguishable from the feature at the center. When the difference in lambda reaches this threshold, then no intensity adjustment can make the feature enter this ellipse of perceptual resolution. We can work out the math to arrive at this threshold as a function of lambda, and this function's shape is completely determined by experimentally known quantities, and the amplitude of this function is scaled by a single scale parameter here, arising from the overall light intensity and our threshold value to define the uncertainty ellipse. And this one parameter fitting agrees with the non-trivial data quite well. Let's review and examine the bottleneck.
So this just puts things to some real examples, yeah? Otherwise, a little bit too much of a teaching abstraction. So with two examples, one is just to say, okay, how can we tell the difference between two patches of light? And that can be understood by this decoding method, yeah? This method is not a method, it's just theoretically what is the best S given decoding, that's definition, okay? It's like what's the best depth? Do I see the disk in front of the ring or behind the ring? These are all decoded values. In the beginning, you don't know what I'm showing you. I ask you to put on the glasses, and you go, oh, that's it, yeah? Another example is, when people do experiment trying to see, oh, is there something in there, not something in there. So all of these are real examples of visual seeing. When you say, oh, is it a cat or a dog? Or you kind of have an expectation because you're in a neighbor's house, they should have a dog. Then you're more likely to say dog. That's the prior, okay? The prior, because you say, what is a priori? What's the probability could be a cat or dog? Yeah, it will put into your decoding. Somebody, yeah. Yeah, because in physical system, nothing is precise. Whenever you get something, just like when you have an input, you can have a different response. Nothing is precise, yeah? Say the response is not precise. You have the same input. When nothing is precise, you cannot be precise, say. It's just noise in a physical, it's not mathematics. However, the statement is mathematics, yeah? Given R, what is S? If you have this function is there, you always pick the function where the S is the max. Okay. And what is the time? 10 more minutes. Okay. So we get some idea how how this could be used, and yeah. Mm-hmm. Oh yeah. And so the idea is here, in fact, the predicted number is much, much higher. The curves are much, much higher. They scale by 21. It's a lot. So why again? It just, this is how people trying to understand. They notice the same shape. Okay, so the Y is very good. You are ready for the Y. Okay, we're going to talk about this is Y.
Also using Poisson neural responses, in which the actual response R in a trial is a random variable around the mean response R bar, which follows this neuron's feature tuning property. And this mean response is often scaled with input intensity I. The response variance for a Poisson neuron is equal to its mean response, and the signal-to-noise ratio is this one, and we plug in the sigma value, we get this result. And therefore, this signal-to-noise ratio should grow with input intensity I. This is, for example, why the wavelength discrimination threshold is inversely related to the square root of the input intensity. Therefore, the discrimination performance should be better when the input intensity is higher. We recall that in this color discrimination task, we have treated each input patch as being sensed by a giant cone, pulling all the cones covering this patch. Therefore, the input intensity should scale with the size of the color patch, and in fact should also scale with the viewing duration of the visual input, since longer viewing should lead to more photon absorption by the cones. However, experimental data show little dependence of the discrimination threshold on the input intensity, patch size, or viewing duration, as long as the patch is not too small and viewing duration not too short and input intensity not too weak.
Okay, the idea is if I make you do a physics experiment, I say measure the size, length of this table. You measure, you have an error. Okay, you say, oh, this is 6 meters. Then if you have an error, what do? You can measure again. Huh? Measure twice, measure 10 times. Then the error gets smaller and smaller. So the idea is when you can look at this longer, it's like you're measured again, again, and again. Yeah? Or when you have this patch very bigger, it has lots of sensors. It's like measuring again, 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 again. And so if you have each measurement give you x1 is the first measurement, x2 is first, and xi, then you can, each of them will have a delta x as the measurement error, and then you can get the average across them. And this average will have a much smaller, this is all learned in freshman physics lab, yeah? And so, however, it shows that you look at that longer, this doesn't help. As if it's not trying to pull up. Or you make this bigger, doesn't help. So this experimental data is repeated in different labs to different measurements, sometimes to longer, sometimes short. No. So the brain is not doing well. So that's the idea. Mm-hmm. What? Pulling response. This is just like a cones don't pull responses. What I mean is when you do this thing, you have something called
and in fact should also scale with the viewing duration
statistical equivalence. This pooling is like statistical
Light
equivalence.
viewing should lead to more
That's
photon
in the
absorption by the
thing
cones.
later.
However, experimental data show little dependence of the discrimination threshold on the input intensity, patch size, or viewing duration, as long as the patch is not too small and viewing duration not too short and input intensity not too weak. This also implies that there is a limit on the brain's power to fully utilize the information available in the sensors. So we have seen an overall picture that ideal observers have their visual performance increase with the number of neurons, input intensity, input size, and viewing durations. However, human observers do not do that. And you may ask whether this is because neural response noises are actually correlated. However, the noise correlation in the real brain is far too weak to explain this huge difference between the ideal and human observers. Such low efficiency in utilizing the sensory information reflects that our visual system is limited by the selection stage in this three-stage framework of vision. Input is first encoded in particular by the retina, and this encoding stage has its own limitations. However, the serious inefficiency is in the attentional selection stage in the central brain.
Okay, so I try to make today shorter. So there's some things you can watch in a video, but rather it is very non-intuitive. So give more time for the question and answers. So you say we don't do that. That's experimental fact. We observe. Now we just have to understand it as a scientist. The experimental data said we cannot utilize all information. And how to deal with it is a problem people keep on trying to postpone. And so tomorrow we have one of the first theories trying to look at it. Yeah. Any questions? Why is it counterintuitive? Yeah, we do not know. I don't know why. It must be depend on the algorithm in there. So the algorithm must be built by the hardware in there. Yeah. And so tomorrow we have a theory that does not deal with that, but we're just starting. Most scientists don't look at it. It's just we don't know. Yeah. Just imagine we're running on a brain that runs at 10 watts. How strong is your computer? Not sure, yeah? A little computer. Quite strong. NVIDIA and all these kind of things, kilowatts of the running. And these days when you want to try LLM, train it's just lots of power. They don't have any power in memory, don't have execution, but we have a power limit. Some animals eat a whole day in order to get enough power just to survive. We eat 2 hours, 1 hour a day, and then we just spend the rest of the day trying to spend that power. And 20 to 25% of power is spent on your brain. And so this is the limit. And with this limit, the hardware must be built in certain ways. And we're trying to understand it. Just like as I say, we don't even see, we're blind. That's very non-intuitive, yeah. Okay. We have ideal observer is what? Yeah? No, don't have to be. Ideal observer is just completely calculated from this. And one example is when they're independent of each other. Ideal observer means just calculate from that. Yeah. And then you have the real human response. You say, oh, human is worse than what they have. Yes.
It's a bit weird Because after that you said that the human, in reality, people have the correlation between the neurons of actual humans is too weak to be considered. I found that a bit counterintuitive that our neurons are not heavily correlated.
Yeah, so this is a frontier problem, as I say. Most scientists do not understand. So I am just giving you a hand waving on the brain energy, but it's like if you use this phone, it's already more powerful than your calculator, but it's less powerful than your desktop. And we have a particular one here that has lots of poor performance, and that's just, you can only understand it that way. And we're trying to see exactly, despite this, we are so able to do these fascinating things of doing a PhD or finding these, this is super powerful. Huh? Notice, yeah, you cannot, if you fix it here, you cannot see it, but your V1 is making you go there. Isn't that something powerful too? So you can see our brain is very stupid, our brain is very powerful. This is the interesting mixture combination. Yeah.
But when we say that our neurons are not heavily correlated, doesn't that kind of mean that
People then trying to look at it and they're not heavily correlated. That was the original original guess. Then they look at the neuron, they're not so correlated. So when I make them uncorrelated, it's a very not too bad approximation.
it's a bit weird for me because when I think of neurons, I think of these things that are like heavily connected. Just like if we can look at like a sketch of a neural network,
Yeah.
we see that all the neurons are really are connected with each other and they affect each other in all these different ways. And I would imagine that in the
Yes,
brain.
but what I say uncorrelated is conditionally uncorrelated.
Okay.
So what is the conditional probability? Their mean response, of course, dependent on each other because they all respond to the same input. But this is the conditional probability. That means the fluctuations of response. Yeah? So, one neuron. So, this is the response. One neuron respond like this, another neuron respond like that, okay? And this mean value depends on the input. So this is R1, depends on input. This is R2. But they depend on the same input. So this value, that value, completely correlated. But the fluctuation is uncorrelated. Yeah.
Okay.
So it is the conditional probability here, uncorrelated.
Okay. Thank you.
Good.
Just, well, thank you. In this right tilted bar, if one cannot find it, then I would say that my brain, for example, is not trained on
You don't need
finding
training.
these. My brain is trained on programming. I find commas in a program that are in the false place or
You don't need
an
training.
extra bracket, but
You didn't even know. You probably went through it. As I said, all of my observers, they never got trained. In fact, we don't let people who have already trained on this to take data. They can do it very quickly. Yeah, machine has all the power. But nevertheless, the human observer could just do this, having an egg without chicken. We notice that if you fix it here, you cannot see it. But your eye go to it. Your V1 is so huge, it's powerful. So that's happy to know, no? And and that's why V1 is huge. Occupies 12% of the whole monkey's brain. It's doing this for you. So that means even if you do not see it, if some cars approaching you or tiger approach you, you'll get it. It saves your life. Even though you're not so great at trying to do these kind of tasks, trying to do these kind of tasks, you're not so great. But you are really great on doing these other tasks. Make you survive dangers in this. Surviving such danger is quite non-trivial. Quite non-trivial, yeah.
I have a question. This model is to consider two options, A or B. It can be extended to consider a third or more options. How the response looks like.
You can do experiment more options, but the thing is, in a scientific experiment, you're trying to make it doable. Just like when you do your physics experiment or pendulum, you want a pendulum, friction-free and all these kind of things. Because you have the solution for that, it's easier to do. So in this case, 2 is already nailing down the problem. Padding 3 or 4 does not really give you more insight into the problem. Yeah. Unless it gives you more insight, that's good. Yeah. Good.
Okay, so I think we learned that the brain is not interested in measuring things to 5 sigma of accuracy.
That's right.
Right? And so essentially wants to save the energy that would be needed to do that for doing other things. So this is, I think, the message, right?
Yeah, yeah. So therefore, what is the brain trying to do? It's trying to do things, it's selecting certain things it's not good at in order to do something else it's better at, I assume, given that this is the power it has.
Mm-hmm. Yeah. Any other question? So, yes, so if not, should we go for a coffee break?
Yeah.
Yes, okay, thank you very much.
And we will have
and please, should the students return back the glasses?
That's right, please give back the glasses.
Yes,
And tomorrow
thanks.
you'll use it again. Yeah.
And another thing, we are going to decide on the dinner of the last week today. So those of you who have not answered to the questionnaire, please do today because because we are going to decide. So, okay.